In this video, we will discuss about something in detail about quality control. So before we go in depth, let us just revise what is this one of the specific quality control, that is for glycoprotein. Uh, so what happens in that is that this ribosome containing mRNA that bind to this translocan complex OST Sec61 and ribosome form a complex, and after that the translation again begins, and as soon as the translation begins in the lumen, the oligosaccharyl this dolicol dolicol glycan that contain glucose 3 mannose 9 galnac 2 pp dolicol will attach to the N-glycan signature site of the with the help of OST, and that signature site is NXST. N is asparagine, X is any amino acid except proline, and S is serine, T is threonine. After that, the glucosidase 1 and glucosidase 2 will trim off this two glycans, so that the monoglucosylated glycan will be detected by calnexin or calnexin and calreticulum, that helps to provide them an isolated environment so that they can form their native structure in the isolated environment. At the same time, it increases the residence times in the quality in the ER. And simultaneously this complex will also interact with PDI family, that is uh, PDI family, that helps in acquiring the native disulfide bond of the newly translated protein. After that, if the protein folding is proper, then the third uh, glycan is also trimmed off by glucosidase 2, and this is UGGT, that detects whether the protein that is detached from calnexin or calreticulum that acquired a native structure or not. If they have acquired a native structure, then they will proceed towards Golgi trafficking, and if they does not acquire a native structure, then this UGGT again reglycosylated the glycan, and so that now the glycan, the monoglycosylated glycan, will again detected by calnexin and calreticulum, and this cycle continue for a certain period of time. After that, the mannose trimming will occur with the help of manosidase, so that, and when the number of mannose were reduced between 5 to 7, then it is detected by the ERAD enzymes, and then it is detected by ERAD enzymes and are being transported to the cytosol uh, for producible degradation. This is a general idea about quality control. We will understand in something within this uh, glycan structure that usually binds to the newly co-translated or newly post-translated glycan with the help of oligosaccharide transferase. So it has been assumed that it has been understand that first glycan is been removed by glucosidase 1, and second and third glycan is reduced by glucosidase 2. However, in vivo experiment shows that glucose in vivo experiment in a specific condition shows that glucosidase 1 alone is not able to remove this glycan. Uh, However, in actual condition, it has been able to remove um, removal of monoglucosidase, removal of this first glucose has been observed. So what people have suggested that uh, glucosidase 1 along with the oligosaccharide transferase Sec61 uh, might form a super complex and uh, acquired a certain specific conformation that facilitate the glucosidase 1 to trim off this first glucose, and this glucosidase 2 will uh, function afterwards when the glucosidase 1 has trimmed off its first glucose, so that it can now be detected by uh, or recognized by calnexin and calreticulin for their function. And one more study that has been conducted is the UGGT knockout cells. In that kind of cells, they have found out three different. They have categorized the glycoprotein based on uh, different release rate, and what they have find out that glycoproteins, there are some glycoproteins that are released from calnexin and calreticulum cycle at the same rate as that of wild type. So that could be because of the reason that could be because of the reason that they only required only one time attachment with calnexin and calreticulum to get their native structure. And there are some glycoproteins, whose release rate are high compared to wild type. So for this can be happen with a possibility that this can be happen with a this can be happen when the in healthy condition, that is in wild type, this type of glycoproteins requires multiple cycle to acquire their native structure. However, this time the UGGT is not present in case of UGGT knockout cells, so they will not be able to uh, re they will not be able to uh, retransfer it to the calnexin and calreticulum cycle. So in that case, the this kind of glycoprotein will also pass in the after the first cycle, either to the cytosol or to the Golgi trafficking, and uh, depending on the abundance of these proteins within the ER, and it will also affect the final efficiency in terms of their native structure, in terms of the native structure of these kind of glycoproteins, as it will all reduced in case of UGGT knockout cells. And third one is the prolonged association of glycoproteins with calnexin and calreticulum, not cycle the with calnexin and calreticulum protein. So this could be because of the reason that this disulfide isomerase F15 usually form a 1-to-1 ratio with UGGT. So in the absence of UGGT, this F15 will become excess and could interact with calnexin and calreticulum, along with the protein disulfide isomerase that usually interact ERB57 to get acquired their native structure. And this SAP51 additional interaction could be the reason for the prolonged interaction of protein with calnexin and calreticulum. So these are the studies that are being done to identify the effect of UGGT on effect or the function of UGGT in the cells. That's all for this video. Thank you.